Hi. In previous sessions, we have discussed the media components, especially carbon sources and nitrogen sources. In this video, I am going to talk about other media requirements. First, we have water. Water is an essential commodity in any industry and especially in bioprocess industry. Water can be used for many purposes, for heating, especially to generate steam and everything. It is used as a cooling agent in the fermenter. We need to cool down the fermenter, and it is used for rinsing, cleaning, and fermentation. The quality of water used for different purposes vary, and some industry need pure water, some other industry don't need that much pure water. So for any industry, we need water in large quantities, and the quality of the water should be consistent. So it should not have very variability, huge amount of variability. In this session, I am going to talk about the use of water for fermentation as a media component, how we are using water. So as a media component, as I told before, we need consistent quality of water. The composition should not change very much. It should be somewhat constant. And depending on the type of fermentation, the quality requirement of the water will vary. Some of the fermentation need purest water, others need just tap water will be fine. If you compare the fermentation done by the or the bioprocess technology using enzymes, or sometimes we are using animal or plant cells, need highly pure water, the milky water, something like that. Maybe deionized water, everything we are using. But for fermentation by yeast or some bacteria, may not require that much purity of water. So sometimes we use tap water. Tap water is cheaper, easily available. You don't have to treat or things like that. It is cheaper, but it cannot be used for all type of fermentation. Sometimes we need soft water. The ions are removed, but it is expensive, so it is used only for the fermentation of high value products, not for the low value, high volume products. So depending on the type of fermentation, the quality of water will change, and the cost of the water will also change. Uh, what we have to do before starting the industry, we should determine the water quality in the area where we are going to put the industry. We should have an idea about the water quality, especially we are doing the mineral analysis. What all minerals are in the water? Some minerals are good for fermentation. It will help the bacterial growth or microbial growth. Some other minerals are not, is inhibitory to the growth of microorganism. So it is very depending on the type of fermentation. So before we like starting the industry, we should have an idea about the water quality in the particular area. So mineral contents, it affect the fermentation. It positively or negatively affect the fermentation depending on the type of mineral and the type of fermentation. Then what we look at is like the pH, which is an important component. Sometimes pH will be in extreme acidic or basic region. The extreme pH need to be balanced before we are using it for the fermentation. So that's an additional step and additional cost. So we don't want water to be in the extremes of the pH. Dissolved salt. As I said before, some of the salts are good for fermentation, but most of them are not that good. Another thing is effluent contamination. The effluent generated by the industry may contaminate the groundwater. So that's a cycle. If the industry is contaminating the water bodies, it eventually contaminate the water sources for the industry. So industries should have the proper wastewater treatment system. Otherwise, the effluent will contaminate the sources. So then sometimes the water acts as a source for trace minerals for the microorganism, especially the micronutrients, which is required only in very small quantities. So once we analyze the components of the water, and if you are satisfied with the water quality, before use we may need to remove the suspended solids, colloids, and microorganisms. These things usually present in the water. If you are taking the water from the rivers or something like that, suspended solids will be there, especially during the time of rain and everything. Even the groundwater from the wells, open wells, have the suspended solids. Colloids must be there, and microorganism will be there. So sometimes we need to remove the microorganism either using sterilization or filtration or simple chemical treatment like ozone things like that don't stay in the water. Ozone is a gas, so once we pump the ozone into the water, it will kill the all microorganism, but the ozone gas will escape from the water eventually. So we need to have some methods to remove of suspended solids, colloids, and microorganism. Sometimes we don't need to remove the microorganism if the fermentation is resistant to contamination, but some other time, so if we are making like for some pharmaceuticals, we should remove the microorganism. Then, for in case of hard water should be treated to remove salt, such as calcium carbonate, which used to be an inhibitor for the microbial growth, and it may contaminate the final product. So, we should remove the salt from the hard water, and iron and chlorine. Some water from some area, especially from the wells or the bore wells, may have the iron and chlorine in it, which require removal. Otherwise, will be carry over to the product, which is not good. Sometimes we need to add salt to the water to control the osmotic pressure and everything. So it is basically depending on the quality of water. Sometimes we need to remove salt from it. In the other case, we need to add some extra salt or some minerals to it. So it's all depending on the quality of water. In case of plant and animal cell culture, as I told before, it needs super pure water. The purity of water should be at the maximum. Sometimes we use the milky water, the minerals and everything is removed from the water, so that pure water we need. That water should be sterile. I mean, completely sterile. It should not have microorganism, and it should not have endotoxins, and it should not have viruses. So we need super pure water for plant and animal cell culture. But other microbial culture, we don't need that much pure water. And another strategy is recycling and reusage of water. Most of the industries, especially bioprocess industry, used to reuse or recycle the water. Especially the water we are using for the cooling purposes, we can use for some other purpose. So we can recycle to another purpose. Or the reuse is possible, especially the steam water. The steam is used for the heating, so the steam can be reused again and again. So recycling and reusage is recommended for bioprocess industry as it requires huge amount of water. So the reuse and recycle will minimize the water cost, and it reduce the volume required to the wastewater treatment. So we are not generating wastewater, so the cost of wastewater treatment will also reduce. So recycling and reusage is always recommended. Another requirement is minerals. Minerals has different functions in bioprocess industry and fermentation. It is used for the osmotic balance of the media. If the medium osmotic balance is maintained by minerals, and it also has the buffering capacity. Some of the minerals, especially the phosphorus, has the buffering capacity. It will control the pH, and it acts as a cofactor for the enzymes. Some of the extracellular enzyme, and in some cases for the intracellular enzymes, we need cofactor to be provided. So the minerals are the cofactors for these things. And some microorganisms may have the special requirements. Some of the fermentation we need to add special nutrients, special minerals along with the media. For example, calcium is used for the streptomycin production. And the thing is that when we are adding calcium, it will precipitate inorganic phosphate. So inorganic phosphate is inhibitory for the streptomycin production. So addition of calcium is essential for the uh, streptomycin fermentation. So if you add calcium, phosphate will go and it will improve the streptomycin production. So those type of special requirement depending on the type of fermentation is there. So those requirements should be met uh, in the medium. So minerals can be a micronutrients or a macronutrients. The macronutrients like magnesium, potassium, calcium, uh, sulfur, uh, calcium, and chlorine. These things need to be provided in high quantities, and most of the times it will be there in the carbon source or nitrogen source. If you are using corn steep liquor as a carbon source uh, or nitrogen source, it will already have huge amount of this type all type of minerals. If you are adding yeast extract, it has all the minerals, so we will get it from there. Sometimes we need minor the micronutrients like cobalt, copper, iron, manganese, molybdenum, or zinc. These things will be in the impurities. Impurities means sometimes it will be in the water, and in some other cases it will be in the media components. So most of times we will get all the minerals from the media components and the as a component or as an impurity. But some cases we need to add it separately. Another thing we need to understand is the chelators. Chelators, we know it used to prevent precipitation during autoclaving as metal phosphate. If the compounds like iron, calcium, manganese, or zinc is present, it may be precipitated as the phosphate, like iron phosphate or calcium phosphate, things like that during the time of sterilization, which is not good because once it is precipitated, it won't be available for the microorganism. So we add chelators. Chelators will prevent the precipitation of these minerals, which is and so as it is not precipitated, it will be available for the microbial growth. Examples for chelators including EDTA, polyphosphates, and citric acid. Though these things are also added to control the precipitation of minerals. And the most common sources of these minerals are the yeast extract or peptone. As I said before, if the microorganism has a special demand or a special requirement of a mineral in high quantity, we need to supply externally. Otherwise, in most of cases, the minerals will be present in the components of the media or in the as a main component or as an impurity. Next, we have the vitamin and growth factors. Uh, Vitamins, like most of the cases, the bacteria is able to produce all the amino acids, vitamins, and growth factors. They are actually self-sufficient. But in some cases, they are not, especially in the case of animal cell culture or the plant cell culture, we need to provide everything. So the vitamins and growth factors including amino acids, fatty acids, and sterols. So especially if you are culturing a animal cell for the fermentation and the bioprocess, we need to provide the amino acid. All the essential amino acids should be provided. All the amino acids should be provided, especially the essential amino acids. Fatty acids like omega-3 fatty acids are essential fatty acids, so that should be provided. The cells are not able to produce them, so we should provide. Sterols will be there. The special sterols like cholesterol or things like that should be provided in the medium. Beyond that, the growth factors like growth hormone should be provided for the animal cell culture. Then only the cells will grow, otherwise the cell will die. So those type of requirements are purely based on the type of organism and the type of process. In case of bacteria, they are able to synthesize almost all vitamins and they don't need any special growth factors for the growth. So most of the bacteria are self-sufficient. We don't have to provide anything. But some of them are. Some of them need some amino acids or some vitamins or some special growth factors for the growth and especially for the production of the products. And filamentous fungus and yeast may need supplements in the medium. We can give these supplements, the amino acids or fatty acids, either in the pure form or it will be present in the plant and animal extract. So plant and animal extracts will be used as a component of medium to get these growth factors and vitamins. And most of the natural carbon and nitrogen source contain most of the vitamins and amino acids, so we don't have to supply. But sometimes the bacteria need it in huge quantities. It may require in more than that percent in the carbon and nitrogen source. In that case, only we need to provide extra. Otherwise, microorganism doesn't need extra supplementation. Next is buffer. Buffering is important. pH need to be maintained. Different type of buffers are used in bioprocess. Calcium carbonate buffer, which is used to control the pH at 7. The other thing is that media components, carbon and nitrogen sources, which contain amino acids, peptides, and proteins, those things have the buffering capacity, the biological buffering capacity. So the amino acids, peptides, and protein will also help to maintain pH. So we should have a buffering system in the medium, like calcium carbonate. Plus, the carbon and nitrogen source also give the support the buffering system. Sometimes the pH of the medium may go beyond the buffering capacity of the medium. In those cases, we'll add acid or alkali to control the pH, especially if the product, if we are making the acetic acid is an acidic compound, so it will reduce the pH. In those cases, the buffering capacity of the medium is not enough to maintain pH, so we need to add it as an external supplement. Or sometimes we are making an alkali, so that also leads to the accumulation of the basic components and increase of pH. In those cases, we will add acid or alkali to control pH, just the way we are using in the fixing the pH of the solutions. So that's all for this session. Thank you so much.